biowaviers, additional considerations for biowaviers, different approaches, how to obtain the biowaviers. There are some exceptions. What are those? We can learn. And we will try to understand these learnings with few case studies, and we conclude the session. Now, starting with what are biowaviers? Now, you know that every year for any regulatory agency, innovator comes up with 18 to 26 NCEs, that is new chemical entities, which get approved as NDAs or the new drug applications. Whereas every year, there are thousands of ANDAs, that is abbreviated new drug applications, submitted to the regulatory agency for getting an approval. If you compare the difference between NDA and ANDA, for NDA, you need to perform or the preclinical studies like phase 1, phase 2, phase 3, then phase 4. But for an ANDA, there is no need of conducting phase 1 and phase 2 studies. And directly you start a large-scale manufacturing by proving bioequivalent to the already existing reference product. So unlike NDAs, for approval of an ANDA, the generic formulation must be proven bioequivalent to the already available reference listed drug. That's why you see that only 18 to 26 NCEs are approved every year, whereas thousands of ANDAs are approved every year. If your drug product has multiple strengths, say for example 8 strengths or 12 strengths, then there is a question that whether you need to perform bioequivalent studies on all the strengths, or I can just do on the highest or the lowest strength and claim waiver, that is waiving of the bio studies for other strengths. So to answer this question, the regulatory agency came up with the concept of biowaiver. So the regulatory acceptance of in vitro testing as a reliable surrogate for an in vivo BE study is commonly referred as biowaiver. So in simple terms, it's usage of in vitro dissolution data instead of in vivo bioequivalence testing. Usually when we say in vitro, most of the cases it's the dissolution testing. So you're going to prove similarity factor calculating by the similarity factor, you are proving that your dissolution profile is similar to that of the RLD or the higher strength of your respective in-house product. So comparison between the test and reference. So you need to do bioequivalence study on the usually on the higher strength, and you can claim a biowaiver for the lower strengths based on similar in vitro dissolution testing. Now, what are the advantages with obtaining a biowaiver? It helps in circumventing expensive and sometimes unethically questionable human testing. Now, because you know that bioequivalence study is being carried out on healthy human volunteers, so a lot of ethical clearances are required before you initiate the study. So you can overcome these unethical testings by using biowaivers. Reducing time in bringing product to the market. So when you plan to do bioequivalence testing on all the strengths, then you are consuming a lot amount of time. At the same time, lot amount of capital or the money is being invested. So biowaiver is advantageous. It reduces the product cost because you are not doing BE on all the strengths. Opportunities for line extensions. Like example, today a new drug is approved with 100 mg strength, but later after maybe 1 or 2 years, there may be an additional new strength coming into the market with a special indication like for new patient population. Especially you see that lower strengths usually arrive for pediatric patients. So in such case, you get extension. So you have pediatric exclusivity. Along with that, you also get an opportunity to have a waiver. No need to do additional BE studies. Just prove the dissolution is similar to higher strength and you can enter the market. post-approval changes for both innovator and generic drug products. So many a times when you start commercializing your formulation, you face a lot of challenges when you go for commercialization. So in such cases, if you slightly alter any changes in the manufacturing formula, manufacturing process, batch size, or manufacturing site change and equipment change. So with the help of certain guidances available, if you are within the guidance recommendation, you can still claim a biowaiver. So we will understand what are these in upcoming slides. Now, there are certain additional considerations for getting a biowaiver approval. First is the excipients. Now, the excipients, whatever you choose in your formulation, should be consistent with IR and MR drug products. Example, if you are using any excipient which has a role in immediate release dosage forms or an excipient with modified release dosage forms, it should be well published, well studied, and there should be sufficient literature to state that this particular excipient acts as an immediate release drug product. This can be used in particular formulation which has the functionality of immediate release or extended release. Now, the excipients that might affect the bioavailability should be qualitatively and quantitatively same in the test and the reference product. Example, the excipients like sorbitol and mannitol, because they are well known to alter the gastrointestinal motility, if your reference product has any of these excipients, then it is recommended to use the same concentration of excipients as used by the innovator in order such that it will not alter the absorption phenomena. Similarly, surfactants like sodium lauryl sulfate, because these tend to alter the membrane permeability. So the concentration of the surfactant in your formulation should be at least similar or can be lower than the innovator. In such cases, granting a biowaiver would be easier. If you go with higher concentrations, there may be a challenge in getting an biowaiver approved. Prodrugs. If your drug product is a prodrug, then there are certain criteria. If the conversion to an active metabolite occurs prior to the intestinal permeation, the permeability of the drug should be determined. That is, actual prodrug has to be determined. If it is after intestinal membrane permeation, then the permeability of the prodrug should be measured. So many a times when there are biorecommendations given, along with inactive, you also measure the active metabolite. Example, I just remember now olmesartan medoxamil. Olmesartan medoxamil, an antihypertensive drug, is a prodrug which gets converted by ester hydrolysis to active metabolite olmesartan. So when you are going for B study, you need to measure the prodrug, that is olmesartan in plasma. And you have to understand whether your drug substance exhibits a linear pharmacokinetics or a non-linear pharmacokinetics. If linear pharmacokinetics, usually higher or lower strength will be investigated for the
Wow, that's true.
Is there any question? So if linear pharmacokinetics, usually a higher or lower strength will be investigated for B. If a drug substance exhibits non-linear pharmacokinetics, then normally higher strength will be investigated for the B study. Usually most of the regulatory authorities indicate on which strength the B study has to be performed. I have certain examples where I will show you in what cases we go for lower strength B study and in what cases we can go for higher strength. If different esters, ethers, isomers, mixture of isomers, complexes or derivatives of already approved active ingredient are used, then they are not eligible for biowaivers. recent example was desvenlafaxine succinate, which was already approved from the innovator, but an ANDA applicant made another salt form of desvenlafaxine, which was not granted a biowaiver since it was completely a new salt or a ester. Hence, they denied the biowaiver. Then the ANDA applicant had to do again the BE study and prove that even a novel salt or ether or ester behaves similar to that of the originally approved drug substance. Now, what are the different approaches for biowaiver? Now, biowaiver can be granted in 5 different approaches. First, for a specific dosage form. Second, when your drug product has multiple strengths. Third, it can be granted based on biopharmaceutical classification system. There is a possibility of scale-up and post-approval changes during your commercialization. If you do certain changes, if well within the SUPAC guidances, you can claim a biowaiver. And there is something called as bridging study. So in these 5 criteria, you can approach a biowaiver. Now, the first thing is comparison. So I have just considered few of the major regulatory agencies who all accept these sort of biowaiver applications. So if you look at this particular slide, specific dosage forms and additional strengths are accepted by almost all the regulatory authorities, which I have listed here, like Australia, Therapeutic Goods Administration, ANVISA from Brazil, Health Canada, EMA, CDSCO from India, US FDA, and WHO. So these 2 accept specific dosage form biowaivers as well as additional strength biowaivers, and also SUPAC-based biowaivers. But if you look at the BCS-based biowaivers, except Japan, PMDA do not appreciate or encourage BCS-based biowaivers. Then there is something called as bridging studies, which is accepted by Australia, Canada, and WHO. Now let us understand each one in detail. Biowaiver based on specific dosage forms. Now this is particularly applicable for oral solutions, parenteral solutions, or locally applied and locally acting solutions such as eye drops. Due to certain characteristics of formulations, the test and the reference product are presumed bioequivalent without further in vivo experimentation. This is possible only in cases where your drug substance, when mixed with ingredients and prepared, it forms a homogeneous solution, which is clear, and there is no need of estimating any further testings. So in such cases, there is no need of performing any bioequivalent study. You can just claim biowaiver by proving that the other physicochemical parameters or the quality target product profile of your test product is matching with the reference product. So the major requisite for this is the excipients are not known to influence the bioavailability. Example I said earlier, like mannitol, sorbitol, or SLS, or any other excipient with a known functionality, and already there is evidence that it alters the bioavailability or alters the cell membrane permeability. In such cases, if you are using those excipients, you cannot claim a biowaiver. If none of the excipients used in your formulation are proven that they do not alter any bioavailability or any other parameter, then in such cases, you can claim a biowaiver. So the biowaiver of this sort is applicable in most of the countries. So here are some examples which I have considered, like bimatoprost topical solution, ofloxacin ophthalmic solution, daptomycin powder, and intramuscular solution of fulvestrant. So if you look at one particular example, the first option, these are the guidances from the US FDA website. product-specific guidances or the recommendations from the FDA. The active ingredient is bimatoprost. The dosage form is solution or drops, which can be used for topical purpose. Strength is 0.03%. Recommended study. Request for waiver of in vivo bioequivalence study. So they directly state that you can claim a waiver. So they completely give a detailed instruction. Again, if you want to claim a waiver, how you can do it. So most of the cases, since we try to be similar to that of the RLD, then you can easily get a biowaiver. So these are biowaivers based on specific dosage form. Now let us look at biowaivers for additional strengths. Additional strength can be a higher strength or it can be a lower strength. Now what cases we use a higher strength and lower strength, we will understand. So if a generic product which you are developing has several strengths, it can be sufficient to demonstrate bioequivalence versus the reference product only with 1 or 2 strengths, depending on certain product characteristics. So if you have multiple strengths, perform BE on 1 or 2 strengths and claim biowaiver for remaining strengths. Now there are 2 approaches under biowaiver for additional strengths. One is look-alike or pseudo-dose proportional formulation, and one is dose proportional formulation. And the conditions for this biowaiver are identical in most of the countries. Now let us take an example. What is first try to understand what is pseudo-dose proportional or look-alike formulation. In this particular example, you have a drug product with 2 strengths, 10 mg and 5 mg. Now, if you look at the tablet weight of 10 mg, it's 100 mg tablet weight. 5 mg tablet has a tablet weight of 100 mg. So whether 10 mg or 5 mg, the tablet weight remains same. So this sort of approach is called as pseudo-dose proportional, or also in simple words called as look-alike formulations. Because different strength, but the tablet weight, size, shape, everything will be similar. Maybe differentiated with color, but rest of the parameters remain same. So this type of dosage forms are called as look-alike formulations. So the drug product is in the same dosage form, but in different strength. The difference in the total tablet weight and size of different strengths is minor. But in this case, if you look at the percentage weight by weight composition of the formulation and compare with the lower strength, only whatever the 5 mg API you have removed from this, you have added to the excipient, that is the mannitol. So 5 mg excess of API when compared to the previous strength, you have added to mannitol. So what happens? Only the slight incremental change has happened to the mannitol. So from 68 to 73. So what you are supposed to do when you are calculating for biowaiver is you need to calculate the percentage difference in the percentage weight by weight composition of the BE strength versus the waiver strength. So here mannitol is 5% difference, remaining MCC and magnesium stearate, it's 0% because both the strengths have 20% and 2%. No need to calculate the difference for the API. So API, you are not supposed to calculate, only the excipient changes, you need to calculate the percentage difference. So the total difference when compared with 10 mg and 5 mg is 5%. So based on this, you can claim a biowaiver. So active and inactive ingredients are not in exactly the same proportional between the different strengths, but the ratio of inactive ingredient to the total weight of the dosage form are within the limits as defined by the SUPAC-IR and SUPAC-MR guidance. Now, when you can say that if your percentage difference is less than 5%, I can claim a waiver. If it is less than 10%, whether I can claim a biowaiver, or if it is more than 10%, what I need to do. So in such cases, to claim a biowaiver, you have to refer these guidances published by US FDA called as scale-up and post-approval changes for immediate release drug products and scale-up and post-approval changes for modified release drug products. I hope you have understood this particular concept of pseudo-dose proportional or lookalike. So irrespective of how many strengths, even it may have 5 strengths, but all the 5 strengths will have the same tablet weight. Only the excipient concentration changes because you are adding the excess amount of API to the compensating excipient. Like usually, most of the times it will be diluent. You don't add excess amount of the quantity to a binder or a disintegrant because you try to maintain rest of the major functional excipients same. Only to increase the bulk of the tablet, you add it to the diluent. Now look at the example for dose proportional formulation. Now here is example for 40, 20 and 10 mg strength. So 3 strengths. So what we do in this case, look at the tablet weights. 40 mg is 400 mg, 20 mg is 200 mg, and 10 mg is 100 mg. As the dosage strength is reducing, your tablet weight is reducing accordingly. So in such cases, what happens? I can prepare a common granules for 400 mg tablet weight, and I can compress to lower tablet weight of 200 mg and 100 mg to get 20 mg tablets and 10 mg tablets. But in previous case, for each strength, I need to prepare separate granules. So for 5 strengths, 5 different granules. But in dose proportional case, we just prepare one common granules and distribute to the lower strengths. So that will make our life easier. So all active and inactive ingredients are exactly in same proportion between different strengths. If you look at the percentage weight by weight between 40 mg, 20 mg and 10 mg, the percentage is no change. So the percentage difference is 0 because it's common granules. So hence you can do a BE study on 40 mg and claim a biowaiver for 20 mg and 10 mg strength by just proving the in vitro dissolution similarity. Example. So here I have taken example of Selexipag and Olmesartan medoxamil. Now Selexipag, because it comes in 8 strengths. Now if you look at here, waiver request for in vivo testing. So it comes in strengths of 200 microgram, 400, 600, 800, 1000 microgram, 1200 microgram, 1400 microgram, and 1600 microgram. So the FDA recommends do the BE study on 1.6 mg, and you can claim a biowaiver for remaining all 7 strengths based on certain criteria. Now, what are those criteria? Acceptable bioequivalence study on 1.6 mg strength and proportionally similar across all strengths. The first criteria is you have to prove that 1.6 mg of in-house product is similar to that of the reference product by conducting the bioequivalence study. So once you prove that you are bioequivalent, then look for whether your formulation is proportionally similar across all strengths. Like previously I said, if you have a common granule, from 1.6 mg you prepare granules and you distribute to the remaining lower strengths, then you are proportionally similar. But also, we just saw the example of 5%, but in US you can go up to 10%. So if you are, the percentage difference between each of the formulation when compared to the BE strength is less than 10%, you can still claim a waiver. And acceptable in vitro dissolution testing across all strengths. So all the dissolution in multimedia, especially recommended from FDA, is pH 1.2, 4.5 and pH 6.8 because it usually the GI pH range would be in this particular range. So FDA recommends at least to do the dissolution in 3 different dissolution medias with an aqueous pH of 1.2, 4.5 and 6.8. So you have to do the dissolution of 1.6 mg and you have to compare in-house product 1.6 mg with 0.2, 0.4 and rest of the strengths. So similarly, I was talking about olmesartan. In US, it is available in 3 strengths, 40, 20 and 5 mg. So you have to do the BE on 40 mg and claim waiver for 20 mg and 5 mg. So the remaining criteria should remain same for all the dosage form, all the drug products, like acceptable BE strength, proportionally similar across all strengths, and acceptable in vitro dissolution testing. I told in previous slide, in what case we can go for a lower strength doing a BE study on a lower strength and claiming a bio waiver for higher strength. Here is an example for lurasidone hydrochloride, which is an antipsychotic drug, which comes in 5 strengths, 20, 40, 60, 80, and 120 mg. Now here they recommend to do a BE study on 40 mg strength, that is the mid strength for this, and you can claim waiver for even higher strength of 120 mg. So usually considering the safety and tolerability issues, because when you administer this lurasidone hydrochloride to a healthy individual, more than 40 mg will lead to safety concerns. Hence, it is not recommended in healthy volunteers to perform the BE study above the 40 mg strength. So maximum recommended strength is 40 mg. So in such cases, you do a BE study on mid strength or a lower strength also is possible. So do a BE on 40 mg and claim a waiver for higher as well as lower strengths. Now, after initially it was only FDA providing the bio recommendations, but of late since 2018, European Medicine Agency also started providing the bio guidances. So on which strength you need to do the BE study and which you can claim a bio waiver. Now here is an example for aliskiren film coated tablet 150 and 300 mg. So they write first it's a BCS class 3 drug. Now what sort of bioequivalence study designed to be selected? Single dose, crossover in vivo. It has to be done on healthy volunteers. You have to do fasting and fed sort of study, and the recommended strength is 300 mg. What is the analyte to be measured? The parent analyte to be measured, that is aliskiren in plasma. Enantioselective analytical method, no. So each and everything detail would be provided in these guidances, and assessment would be based on pharmacokinetic parameters like AUC and the Cmax, and 90% confidence interval should be between 80 to 125 to meet the bioequivalence criteria. So this is just to show that how, on what basis we select a particular strength to do the BE. So most of the regulatory authorities give a recommendation on which study can be performed. In cases where you don't have any available literature, you can always consult. You can write a controlled correspondence to the regulatory agencies to understand which particular strength can be selected for a BE study and whether can I claim a waiver for the lower strengths. And they usually take 1 month to reply to your query. So you have to wait for 1 month, you get the response. Based on the response, you can plan your study. Now coming to the third approach, BCS-based biowaivers. Now, I'm pretty sure you all know what is BCS, its classification, and what are the criteria to classify a drug substance as BCS 1, 2, 3, or 4. So in regulatory perspective, a complete biowaiver is possible, that is, without doing any BE study, neither on higher strength. Don't do any BE study, just claim a biowaiver. It is possible if it's a BCS class 1 or class 3 drug. But there are certain criteria and varies from different regulatory agencies. Now let us look what are the different criteria for BCS-based biowaivers. Now, you know, according to the BCS, a drug substance is classified as high soluble if the highest dose of the drug substance is soluble in 250 ml or less of aqueous media over a pH range of 1 to 6.8. And if your drug product has availability or bioavailability of more than 85%, it's considered to be highly permeable. The rationale behind 250 ml, it's the volume which is derived from typical bioequivalence study protocol. So whenever any clinical studies are being performed, it is usually understood that a human usually takes the tablet or any dosage form, usually solid oral dosage form with 250 ml, that is 8 ounces of water. Hence, based on that protocol, 250 ml is selected as the criteria for classifying the drug substance as high or low soluble. Now let us look in different regulatory scenario when you can say it as high soluble or low soluble, or what are the criteria to test a drug substance. So remember here, I would not be talking about Japan because Japan does not accept BCS based biowaviers. So Japan we can ignore for the time being. If a drug substance with the highest dose strength, you have to perform the solubility studies, and the volume of solubility testing should be less than or equal to 250 ml. So almost all the regulatory authorities accept this criteria to classify a drug substance as high soluble. So volume should be less than or equal to 250 ml. And usually to say high soluble, you have to consider the solubility parameter of the highest strength. Now, some regulatory authorities require you to do solubility testing in pH 1 to 6.8. Some of them recommend 1.2 to 6.8, and some of them like 1 to 7.5. In this case, India recommends performing solubility testing in pH 1 to pH 7.5. WHO, Health Canada, and Anvisa say pH 1.2 to 6.8, whereas FDA, TGA, and EMA recommend 1 to 6.8. So you have to do the solubility testing within this pH span. And additionally, to classify a drug substance as high soluble, you have to do solubility testing at pKa +1 and pKa -1. Now, which are the countries which require this testing? pKa dependent. So if your drug has a pKa of example 3, then you have to do solubility testing at pH is equal to pKa +1, that is 4, pKa -1, that is 2. So you have to do testing at 2, 3, and 4. Now, US FDA, Europe, and Australia recommends you to do this, pKa ±1, whereas rest of the countries do not have such criteria to test the solubility in. So you have to estimate the concentration at saturation, that is saturation solubility testing. Temperature is 37 ±1 degrees centigrade. Usually, most of them recommend duplicate, solubility testing in duplicate or triplicate. Europe and Australia can accept duplicate testing, but remaining, it's triplicate. And the method for solubility testing is shake flask method is the most commonly used. If you are using any other method, then you have to give a justification for using other than shake flask method to determine the solubility. Now, to classify your drug substance as high permeable, most of the regulatory agencies accept more than 85% of systemic bioavailability. Many a times in the product label, if you go through the label in pharmacokinetics section, you will see the absolute bioavailability or the permeability data. They'll say that more than 85% is absorbed or more than 95% of the drug is absorbed. In such cases, with the product labeling evidence, also you can claim a drug substance as highly permeable. Only for US, acceptance of literature data, if there is no information in the drug product label, but if it is available in literature, some review or research papers published in the public domain, except US would not agree the published paper information, but instead they want you to perform an additional testing. But remaining regulatory authorities accept the literature data as well. If human data like absolute bioavailability or mass balance using biopharmaceutical drug disposition classification system is available, then it is widely accepted. CACO2 cell permeability studies is accepted as primary data only in India and USA. To prove that your drug substance is highly permeable, as a primary data, only FDA and CDSCO will accept your application as a high permeable drug. If it's a primary data for remaining markets, just with CACO2 permeability, they would not accept. It can be a supporting data, but not a primary data. Like for remaining of the markets, it's not considered as a primary data. So the permeability studies can be performed using absolute bioavailability studies, mass balance using biopharmaceutical drug disposition, in vivo perfusion studies, in vitro excised human or animal intestinal study, or in vitro epithelial cell culture study. So these are the different techniques which you can use to prove your permeability of your drug substance. Next is in vitro dissolution testing. Now we know that BCS, first was the solubility, second was the permeability. Then when we are going to claim a biowaiver, in vitro dissolution of the dosage form. So most of the agencies expect you to perform dissolution testing in at least 3 medias, that is 1.2, 4.5 and 6.8. The volume can be 900 ml or less than 900 ml is accepted. But surfactants are not accepted because you're claiming it's a BCS class 1 or a class 3 drug. Hence, you cannot use any surfactants. Enzymes are accepted for gelatin capsules or tablets with gelatin coating for most of the agencies except USA. USA would not accept a waiver if you are having gelatin capsules and you're adding enzymes in the initial stage itself. If for your release of your drug product, you are adding an enzyme, then it is not considered for biowaiver in US, but remaining markets accept it. Usually on stability, if you see such sort of behavior or phenomena because of the phenomena of gelation of the capsules, that is due to aldehyde linking, you see something called as pellicle formation. In such cases, you can add an enzyme and prove that it is due to the pellicle formation. But for remaining markets, they accept as such at the release itself. But US doesn't. Temperature for performing the dissolution testing, 37 plus or minus 0.5 degrees centigrade. So 36.5 to 37.5 for India, US and WHO. Whereas for Europe, Canada and Visa and TGA, you can go up to 36 to 38%. So plus or minus 1 degrees. The apparatus, that is the dissolution conditions, apparatus and the RPM. Paddle at 50 RPM is widely accepted. Basket at 100 RPM is widely accepted. Paddle at 75 RPM is accepted easily without any further questions from WHO. But if you use for any other markets, you need to give a justification why you are using 75 RPM using a paddle type of apparatus. Because usually paddle at 50 RPM is expected to have sufficient hydrodynamics. And since your drug substance is a BCS class 1 or class 3, it is expected to dissolve more than 85% in 15 minutes. So hence, higher RPMs are not recommended for BCS 1 and BCS 3. Now, this slide represents when you say a drug is rapidly dissolving and when it's very rapidly dissolving. So usually for BCS class 1 in Australia and Visa, Canada and Europe, it's more than 85% drug dissolved in 30 minutes. It's called as a rapidly dissolving drug. For BCS class 3, if the drug dissolves, because BCS class 3 has a disadvantage of being low permeable. So you have to prove that permeability with the help of in vitro or in vivo studies, and then you can claim a waiver based on rapidly dissolving dissolution behavior. That is more than 85% in 15 minutes is considered to be rapidly dissolving. But here to say for BCS class 1, we say that more than 85% in 30 minutes is high soluble, but usually BCS class 1 drugs exhibit more than 85% in 15 minutes itself. So to claim a biowaiver, you have to do dissolution testing on not less than 12 units. And similarity factor, that is the F2 testing, is the widely accepted method for considering your dissolution is similar to that of the RLD. So alternate methods to F2 can be accepted in few of the regulatory agencies, but Brazil do not. Brazil does not accept alternate methods apart from F2 calculation. Similar classification for India and US as well. More than 30 minutes for BCS class 1 and more than 15 minutes for BCS class 3. Additional criteria for a BCS class 2 drug, if it's a weak acid and if it dissolves more than 85% in 30 minutes, only WHO will accept this application for a biowaiver. But rest of the markets doesn't have this criteria if it's a BCS class 2 drug. Now, exceptions. Even though your drug substance may be BCS 1 or BCS 3, but still there are certain limitations. Example, if your drug substance exhibits narrow therapeutic index, owing to the high risk, it is not eligible for biowaiver. Drugs like digoxin, phenytoin, theophylline, lithium carbonate, carbamazepine. So because these are NTIs, narrow therapeutic index drugs, they're not eligible for biowaiver. Drugs which tend to be absorbed from the oral cavity. Sublingual buccal tablets, which are formulated to dissolve quickly in the mouth, are not eligible for biowaivers because most of the drug substance is dissolved in the mouth and absorbed directly into the systemic circulation via buccal mucosa. Hence, they are not eligible for biowaivers. BCS-based biowaivers have limited application for BCS class 2 and not at all applicable for BCS class 4. So do not think if you have a BCS class 4 drug, you can claim a BCS-based biowaiver. Example for BCS-based biowaivers, pregabalin capsules, lacosamide tablets. So they give you two options. Either you can claim a BCS-based biowaiver, or if you are interested, if you have more money, if your industry has enough resources, enough money, and enough time, then you do the in vivo studies. So most of them go for a BCS-based biowaiver approach, where you just need to prove that the qualitative and quantitative composition of your product is similar to that of the reference product, and all the strengths release more than 85% in 15 minutes or 30 minutes. Then in such cases, then you can claim a biowaiver. No need of doing BE on even higher strength. Directly go for a submission with 6 months of stability data. Similarly, in Europe, for agomelatin tablets, it's a BCS class 1 drug. So you can directly go for biowaiver. Or if you're still interested, you can go for a fasting BE study. Now coming to the fourth approach, biowaivers based on scale-up and post-approval changes. In short form, we will be calling it as SUPAC. So during product lifecycle management, there are several post-approval changes which may require a new BE study. As I said, during commercialization, you will start facing certain issues. Example, sticking or picking, or you will fail to meet the expected dissolution criteria. In such cases, you cannot go back and do the complete R&D. So in such cases, you can slightly tweak in your formulation, slightly increase the disintegrant to dissolve the drug faster, or increase the lubricant quantity to avoid sticking and picking. So a slight change, 0.25, 0.5% can be acceptable and no need of doing any BE study for that. So such sort of biowaiver, seeking a biowaiver are called as biowaivers for scale-up and post-approval changes. So all these changes tend to happen after approval of your drug product. So when it is in the marketing. So different regulatory authorities have a different opinion on this as well. First, let us look at SUPAC IR, that is the FDA recommendation. Now, if you should remember the example what I had given in the previous slides, example Olmesartan 40, 20, and 10 mg. We looked at those three different strengths and we saw the difference was completely zero. So when it is zero, directly you can go for a waiver. If you remember the pseudo-dose proportional example, where the absolute difference between 10 mg strength and 5 mg strength was 5%. If it is less than 5%, just dissolution release requirements are enough. Just do the dissolution of at the release stage in the release media, that is the QC media, where you need to just have a single stage dissolution testing, just as 30 minutes or 15 minutes, and prove that there is no change with 5% as well. So it is a level 1 change of SUPAC. If you are further increasing any of the excipient and the percentage absolute difference between the two strengths is more than 10%, then it is called as a level 2 change. In such cases, you have to do dissolution at release, plus you have to do similarity testing in different medias, that is multimedia like pH 1.2, 4.5, and 6.8. So in this level 1, it's only the release, only one media. It may be 1.2, or it may be 4.5, or it may be 6.8, or sometimes it may be just water also. But when it comes to level 2 change, in addition to the release media, you need to also perform dissolution testing in multimedia. If your total excipient change is more than 10% to the approved quantitative composition, then it's called as a level 3 change, and you have to do the in vivo bioequivalence study. You cannot claim a biowaiver with change more than 10%. So SUPAC has level 1, 2, 3 changes. Level 1, just release testing. Level 2, release along with multimedia testing. Level 3 calls for a in vivo bioequivalence study, which no industry would prefer to do, because already your product is in the market. You don't want to do the BE study now and again reinvent the whole wheel. WHO, you can consider directly anything less than 10% is acceptable with just dissolution requirements. If more than 10%, calls for a BE study. Now, modified release dosage forms. In this, US FDA has clearly stated two different scenarios again. Non-release controlling excipient. If it is a non-release controlling polymer, especially because modified release, you have a functionality polymer. If it's a non-release controlling excipient, up to 10%, just similarity in dissolution profiles. If more than 10%, BE study. Whereas if it's a release controlling polymer, then if it crosses 5%, you have to do a BE study and similarity in dissolution profiles. So this is the major change which happens if it's a release controlling polymer or excipient. More than 5% calls for a BE study. Less than 5%, you can just do the dissolution testing at release as well as in the multimedia and prove that it is similar. For Canada, anything more than 5% is a BE study. So less than 5%, just dissolution. More than 5% in vivo BE study. When it comes to Japan, Japan is little more liberal on this. Anything less than 5% is no problem. If more than 15%, it calls for a BE study. So up to 15%, you can just do similarity testing and comparative dissolution profiles in multimedias like 1.2, 4.5, and 6.8. If more than 15%, you have to do the BE study. And additionally, if it's a narrow therapeutic index drug, you need to do a in vivo study for Japan. For Europe, Europe is almost similar to Canada, where anything more than 5% change will call for a BE study. At the same time, For Europe, you have to specify that the drug product is manufactured by same manufacturing process. So you have to state that 5 mg, 10 mg, 15 mg, 20 mg are manufactured by using the same manufacturing process in the same site. The qualitative composition of the different strengths should be same. So no excipient change is acceptable. The only big difference what happens in Europe is when you have a combination drug like olmesartan and hydrochlorothiazide, so two active substances. So in Europe, the second active substance is considered as an excipient. So when you calculate the percentage difference for Europe, you have to consider the second API also as a excipient. Whereas for US, Canada, Australia, you can consider it as an excipient, but for Europe, it's an excipient for Europe, not for rest of the countries. So you have to consider while calculating the percentage difference. So during the case study, I would explain a single example with applicable for all the markets. Now, bridging studies. Biowaiver based on bridging studies. Many countries require local BE studies to be done. Example, China. I can say a classic example for China because currently I see here, they will not accept a biowaiver based on bridging study until the BE study is done on Chinese population. Similarly, Japan, Mexico, and Brazil. So the local reference product, the reference product which is available in China, they expect the innovator should have done these studies on Chinese population as well. Then only they can accept a waiver. Until then, they don't. This is due to the ethnic sensitivity relating to the metabolism of the drug substance. It makes sense to demonstrate bioequivalence in different ethnic populations. So ethnicity and the races lead to this study. As innovator products are often very identical because innovator when launched into the market, they make a product for global market. And for innovator, many times it is not possible that he do the BE study in each and every country. So they just do the BE studies in particular races or ethnic volunteers, and then they release the product into different markets. But these countries expect you to do in the local population as well. But there are certain countries like Australia, Southeast Asian countries like Vietnam, Cambodia, Korea, etc., Canada, and World Health Organization accept the biowaiver application irrespective. If you have done in any other part of the world, they can accept that particular reference product data for granting a biowaiver for your product. Now let's understand these approaches with example of a case study. Here I have taken a BCS class 2 drug. It is available in 4 strengths and biorecommendation is for a 20 mg strength. So I cannot say that B study is not at all required because it's a BCS class 2 drug, and BCS class 2 drug until it's a weak acid and I'm going to submit the application for World Health Organization, I cannot claim a biowaiver. I have to do at least 1 BE. So we have to design a biowaiver strategy for USA. So we have to just remember for USA, SUPAC guidance. So you remember level 1, level 2, and level 3. So let us try to understand the formulation first. So 4 strengths, and let us look at the tablet weight, 150 mg. So there are 3 strengths with 150 mg and the lowest strength with 90 mg. So the innovator or in this particular study, so we have made a granules for 20 mg strength and compressed to 150, separate granules for 15 mg, separate granules for 10 mg, and separate granules for 5 mg. So you have to prepare 4 different granules for 4 different strengths, but be only on 20 mg. How do we claim a biowaiver in USA for remaining 3 strengths? Now let us calculate the absolute difference between the 20 mg strength and 15 mg strength. So 20 versus 15. So remember, no need to calculate the API difference. Just calculate the percentage difference in the excipients. So only change is the amount of MCC. Lactose is 0, SSG is 0, and magnesium stearate is 0. So the total difference is 4.23%, which is well within the level 1 change. So if I prove that the release dissolution criteria and in multimedia, if my F2 is more than 50, then I can easily claim a waiver for 15 mg strength. No issues. No need to do a BE study for 15 mg. I can claim a waiver. Now let us look out for the next strength. 10 mg. 20 versus 10. The percentage difference again is changed only in the MCC concentration. So whatever the excess of API was there, you have removed that and added to the MCC. So the percentage difference between 20 mg strength and 10 mg strength is 8.47%. Rest of the excipients are similar. So 8.47 in USA falls well within the level 2. It is above level 1, but meets within level 2, that is less than 10%. So again, just performing in vitro dissolution in release as well as multimedia is enough to grant a biowaiver, provided your F2 is more than 50 in all the medias. Look out for 5 mg. So 20 mg versus 5 mg for USA. The absolute difference again, even though what you have done is you have reduced the tablet weight to 90 mg, but you have tried to maintain the percentage composition less than 10%. So the percentage absolute difference between MCC in BE strength and the waiver strength is 9.88, which is less than 10%. Hence, for USA, you can claim a biowaiver. So just to do the BE study on 20 mg and claim biowaiver for remaining 3 strengths based on in vitro dissolution similarity. Now let us take the same example and understand what would be this scenario if it is for Canada and Europe market. Now let's first look out for Canada. You know that 20 and 15, it's 4.23% difference. So Canada has only 2 levels, less than 5%, dissolution more than 5%, BE. So 15 mg can be waived off the BE because it's less than 5%. 20 versus 10, more than 5%. Hence, you have to do a BE study for 10 mg strength. You cannot claim a waiver. And since I have taken Europe also as an example here, you also remember that Europe also anything more than 5% calls for a BE study. So it's common for Canada and Europe. So you have to do a BE study for 10 mg and you can claim a waiver for 5 mg. So when you do BE study for 10 mg, you have to consider this for calculating the percentage absolute difference. So 10 mg versus 5 mg. The absolute difference again is only the diluent, 1.41%. So you can claim a waiver for 10 mg based on BE study of 10 mg. So for Canada and Europe, it will have 2 BE studies. The same formulation for USA, only 1 BE study and 3 strengths can be claimed a biowaiver. Whereas for Europe and Canada, 2 strengths will go for BE study and 2 only you can claim biowaiver. I hope with this example, it would be much more clear for you. Now let us see one more case study with a BCS class 4 drug. Active ingredient A is a BCS 4 and active ingredient B is a BCS 1. It is available in 2 strengths and biorecommendation is for 40-25 mg strength. Design a biowaiver strategy for US and Europe. So let us look at the tablet weight, 400 mg for both the strengths. So you have to do the BE on 40-25 and how can we get a biowaiver for lower strength. For USA, Remember, for USA, whether 1 API, 2 API, or 3 API, no need to consider API for the calculation. You just need to consider the excipients. And the total percentage absolute difference should not be more than 10% for level 2, 5% for level 1. Now, the percentage absolute difference is 3.125, which is well within level 1. So just do the dissolution. Similarity proving the similarity will get you a waiver in USA. But if the same formulation I want to give for Europe, what happens? The percentage absolute difference is more than 5%. Why? Because in Europe, the active ingredient B is also considered as an excipient. So the percentage difference between B strength and the waiver strength need to be considered while calculating for Europe. So the total difference is more than 5%. Hence, you have to do B study for 4025 as well as 4012.5. So for US, 1 B study, for Europe, both the strengths B study. Here is a BCS class 1 drug available in 2 strengths. So what would be the biowaiver strategy for Japan? So since I have already mentioned BCS class 1, and if you remember, Japan does not accept BCS-based biowaiver applications, so not possible. But if the same was in US or Europe, it is still possible. So with this, I think you would have at least got some information or insight into what are biowaivers, what are the different approaches to get a biowaiver. So to conclude, currently several applications are under review for waiving bioequivalence, because when you reduce the unnecessary human testing, the number of applications would significantly increase, because just performing in vitro dissolution and submitting the application would take a shorter time, and it would be easier for the review by the regulatory agencies. Hence, there are many applications under review for bioequivalence waiving. BCS-based biowaiver has major advantage of cost and resource reduction because it does not have any human testing, it's just dissolution. Logically, small change in formulation should not affect the performance of the drug. So similarity can be proved with in vitro dissolution testing. Because when you're claiming waiver for lower strengths, only a slight change is happening in the excipients, plus or minus 5%, and up to 10% it is acceptable in most of the regulatory agencies. So this slight change is not expected to have a significant influence on the in vitro dissolution. It is expected in upcoming time, the regulatory authorities will consider other classes of BCS for biowaiver study based on risk assessment. Very soon, there is a discussion that even for BCS 1 and 3, they don't want to do dissolution, but instead, just based on disintegration time or disintegration test, they would waive off even dissolution. So that is still under draft and discussions are under progress with different regulatory agencies. The lowering of regulatory burden will result in regulatory relief without loss of product quality, because our ultimate aim is to arrive at a quality product with desirable safety and efficacy profile. So these are the references for this particular presentation. Most of them are the regulatory guidances from different regulatory agencies like FDA, Therapeutic Goods Administration, EMA, and Health Canada, CDSCO, and WHO. So thank you very much for your patient listening. And now the session would be open for question and answers.
Thank you, Zaheer. The session was very interesting. In a very short time, you have compiled the data and we could get each and every aspects. You have given a comparison between the guidelines of the different countries, which was very, very informative and very helpful. I hope the delegates have enjoyed the session. There's one question in the chat box.
Why Japan is not following the regulatory guidelines as others? Yeah. To give a brief overview, Japan has a completely their own set of guidances. It would be typically to hear that Japan people, especially Japanese, don't like to have bigger tablets. They prefer smaller tablets, just like 8 mm or 9 mm, not more than that, unless it's required. And they want all the tablets to be in white in color. They don't prefer tablets to be colored. It's again their individual perception, which we do not comment on. And Japan has a strict regulatory authority and guidances when compared to US FDA or Europe. Japan is much more stringent than other countries. So Japan does not accept the BCS-based biowaiver applications. And if you look at the Japan PMDA website, there is something called as information documents. Japan expects, irrespective of the type of dosage, solid oral dosage form, whether immediate release or extended release, water is a mandatory media for performing dissolution testing. So Japan expects you to do dissolution in water as well, irrespective whether it's soluble or insoluble. Even if it's insoluble, you know from the solubility data, Japan wants to do the dissolution testing for up to 6 hours in water or any other media in which it is insoluble.
There's one more question. Is there any different guideline for biowaiver of a nano formulation?
No, for nano formulations, there are no biowaivers as such right now, because these biowaivers are majorly applicable for oral solutions or any solution, because nano formulation, usually it may be in the form of a suspension, quite difficult for the time being, but later off late, we have to see if there are certain guidances coming up for nano formulation biowaiver approaches. But as of now, for nano formulations, there are no guidelines on biowaiver.
One more question. What is bridging?
Yeah. So as I said, some countries, specifically example I took of China, where it requires the B study or the bioavailability studies to be performed in Chinese volunteers, then that is not bridging. But there are certain countries like Canada, WHO. Example, if a reference product is available in USA market and not available in Canada market. So in such cases, if you want to launch your product in Canada, how do I do? So Canada has an option of something called as bridging. So you can compare your dissolution of your drug product with the US available drug product, prove the similarity and submit to Health Canada that you have performed bioequivalence study with a US-based reference product, or you are claiming a biowaiver based on in vitro dissolution of the USA reference product. Health Canada will still accept your application because they accept foreign reference product also for the granting of biowaiver. Similarly, this was just an example of US and Canada. If you look at Europe, Europe has 33 countries and European Union has divided these 33 into RMS and CMS countries. Now, what is this RMS? RMS is reference member states and CMS is concerned member states. If somebody is an regulatory expert, they would understand. For Europe, there is something called a centralized procedure and decentralized procedure, apart from mutual recognition procedure. Mutual recognition is among these 33 countries, they discuss among themselves, like if a product is approved in Germany without any further BE study, just based on in vitro dissolution similarity, I can launch into France or Spain or Poland. But there are certain CMS. There are 7 CMS as of now. Major are Germany and Netherlands. If a reference product you consider from Netherlands or Germany, perform the BE study or claim a biowaiver based on these, and you can submit your application as a centralized procedure. So once if it's get approved in Germany or Netherlands, you can submit the same in vivo BE study for the CMS countries. If your drug product is available only in CMS, then you cannot market those products in the RMS countries. So that's again a completely different topic where it all depends on mutual recognition between 2 countries. I hope that would answer most of the question on the bridging.
Again, the genetic setup of a people or a population also matters for bridging.
Yes, yeah.
Like we have seen in the COVID vaccine.
Yeah, the efficiency or the efficacy of the vaccine in China, if I talk about the Sinovac or the Sinopharm vaccine, it's in China, they show around 50 to 60%, but the same vaccine in Brazil had more than 70%, and few other countries, it was almost like Turkey had reported almost 89% efficacy of this particular vaccine. So it varies according to the ethnicity of the population.
Yeah. One question, all these guidelines are also applicable for herbal formulation?
No, for herbal formulation, these guidelines are not applicable. There are certain other guidelines. If you look at FDA, like there may be manual of policies and procedures and specific product-specific guidances available, which will deal with herbal formulations. These guidelines are not applicable for herbal.
There's one more question, Zaheer. Is there any impact? Is there any impact on regulatory procedure after the incident?
Brexit, yeah. So since United Kingdom took a decision to get exit from the European Union, so after that, what had happened is Netherlands was considered as a site where all the imported drug products would go for testing. So Netherlands will test the dosage form or the drug product before sending it to the UK market for marketing. So after Brexit, the additional responsibility has been given to Netherlands, that is HMA, Head of Medicines Agency, the regulatory authority of Netherlands, which will be testing all the samples before it moves to UK. So this was not the case earlier when UK was a part of Europe. So once it's got separated, this is an additional entity for Netherlands.
Can you explain the examples of 5 and 10% excipient change in the formulation?
To this, I will go back to my screen.
Yeah.
Well, let us take an example of the case study 1, where we had 4 strengths. Now, if you compare the percentage, as I said earlier, we have to consider the percentage weight by weight composition to calculate the percentage difference. Now, if you look at this example, 20 mg versus 15 mg, no need to consider the API for calculation. Now, the 20 mg tablet has 57.08 mg, which constitutes to 38%. And 15 mg has 63.43 mg, which constitutes to 42.29%. So you need to calculate the difference, 42.29 minus 38.05, that is 4.23. So this is the microcrystalline cellulose difference from 20 mg to 15 mg. Let us look at other excipients. 20 mg also has 40% of lactose. 15 mg also has 40% of lactose. Same case with disintegrant, 4%, 4%. Magnesium stearate, 1%, 1%. So the difference is 0. Now, when you sum all this, the total absolute difference between 20 mg strength and 15 mg strength is 4.23%, which is less than 5%. So if it is less than 5%, for most of the regulatory agencies, you can just prove in vitro dissolution similarity and claim a biowaiver for 15 mg strength because it is well within the level of less than 5%. Now, to understand 10% difference, here I do not have an example, but I will take the worst case of 20 versus 5 mg. If I had this tablet weight of 100 mg. Example, instead of 90, I had 100 mg. So I have to add 10 mg more of MCC. So MCC would become 53.14. So the percentage weight by weight is 53.14 because my tablet weight is 100. So I have to assume now this is 100 mg. So I add the excess 10 mg to the MCC. And to claim a biowaiver for lower strength, what I will do, I will compare the B strength, that is 20 mg with 5 mg. So what happens here? It is 53.14 minus 38.05. So what is the percentage difference it is coming? 15.09. So here, what is the difference of MCC? 15.09. Rest of the things would remain 0. If I'm not going to change, like 40% of lactose, 4% of disintegrant, and 1% maize starch. So to give an example, for more than 10%, I just made this tablet weight to 100 mg. Or if you want me to do online, I can try. So if I make this to 100 mg, I have to increase this to 1 mg, this to 4 mg, and this to 40 mg. So if I do this and I calculate the percentage difference, 53.14 minus 38.05, it comes to 15%. So it's more than 10%. That is more than level 2 change. When it is more than level 2 change, it calls for a B study. So this is one example I could take at this moment for you to explain about 5 and 10%. I hope this will answer your query.
I hope the query has been answered. There's one more question. Can we change the brand of the excipient? Like instead of...
Yes.
Yeah.
The brand of the excipient can be changed. Excipient grade can be changed, but excipient should be similar when you want to claim a biowaiver. If they are using mannitol, because most of the times innovator would not tell mannitol 200 SD or 100 SD, whatever they have used. They just write mannitol. Now it is you have to do the risk assessment and select a suitable mannitol for your formulation, and you can still claim a biowaiver. Only thing is, it should have mannitol. Any grade, any manufacturer, doesn't matter.
Okay. Any other questions from the delegates? For this session, 2 of our resource persons have joined, Tima Shetty sir, as well as Sarsija madam.
Yeah, good evening to Dr. Sarsija madam and Dr. Tima Shetty. I have been an idol for Dr. Sarsija Suresh is an idol for me because I usually follow her every time. It is a pleasure that madam is listening to my lecture today.
Yeah.
Good evening, sir. It was a very nice presentation, sir.
Thank you very much, sir.
Yes, Zaheer, it was a very informative and detailed presentation. Very good.
Thank you very much, madam.
There's one more question, Zaheer, from the delegate. Like same on the brand of the excipient, can we change the type of the starch like maize, potato?
Yes, yes. If your aim is a disintegrant, if you're in your formulation, usually because maize starch can at smaller concentrations can be a disintegrant, but at larger concentrations it can be acting as a binder. So depending on the functionality, what you expect, you can still go for change. Because usually potato starches are usually used as disintegrants. Maize starch can be used as a binder come disintegrant. So it depends on what functionality you're looking for.
So all the messages from the delegates that this was a very wonderful session and you have clarified most of the doubts of the delegates. Thank you, Zaheer Abbas, for your wonderful presentation. I hope there was a last moment change in your schedule because one of our speaker, because of his busy schedule, he couldn't attend today's session. So hence I requested Zaheer Abbas to give one presentation, and last moment sir agreed, and it was a really wonderful session. Thank you so much. I hand over
Thank you very much.
I hand over the presentation to Harish sir to give you the vote of thanks. Thank you.
Good evening, one and all. Good evening, sir. I thank Mr. Zaheer Abbas sir for giving an very informative and interactive presentation on this Biowaviers. What are all the opportunities are there? What are all the requisites are there? And their overviews with the different regulatory bodies. How it nicely has been explained, and the queries from the delegates has been cleared. I think it is cleared properly. With this, I will very thankful for the Zaheer Abbas sir. In a short time, he had prepared it and he has presented in a very good manner. Thank you, sir. Thank you for your efforts and all the your continue your efforts, sir. Thank you, sir.
Thank you very much. It's my pleasure as always. Thank you.
Okay, the feedback form is already put in the chat box. Kindly fill. I request all the delegates to kindly give your valuable feedback. Tomorrow is the last day of our FTP, ATAL FDP. I also request the three resource person to please join at the validatory function. Tomorrow, the validatory function will commence at 3 p.m. I will again I will again intimate you all personally on your WhatsApp group. Please kindly join for tomorrow's validatory function, so that we'll have a valuable feedback from you, so that we can improvise for the next sessions what we'll be conducting at KLE College of Pharmacy, Hubli. This is my kind request. Zaheer, kindly please join. Sarsija
Definitely, I
madam,
would try.
yeah, Sarsija madam as well as Tima Shetty sir, please kindly join tomorrow's validatory session. With this, I end the today's meet. Okay, I request all the delegates to rejoin tomorrow morning at 9:15 a.m. so that we'll exactly start the session at 9:30. Tomorrow also we have a very interesting session in the morning. We have a session by Shri Hemant Narvekar. He's a research scientist, Formulation Development, Unichem Laboratories, Goa, followed by the second session by a very eminent speaker, Dr. Sarsija Suresh ma'am. She'll be speaking about the emerging trends in vaccine drug delivery. So I request all the delegates to make maximum use of these resource person and kindly join the session on time. Thank you. The feedback form is posted in the chat box itself. You can click on the link in the chat box, and personally I will be posting it in your WhatsApp group also. Thank you one and all. I shall end my meet. Thank you, Zaheer. So it was a very wonderful session. Okay.
Thank you. Yeah, good day. Bye.
Good day. Bye.